Hello everyone, hope all of you are doing well. In today's session, we are going to discuss about the structure of prokaryotic cell. Some bacteria, blue-green algae and mycoplasmas are the examples of prokaryotic cell, and they don't have a membrane bound nucleus, or the nucleus of these prokaryotic cells are not well organized. In today's session, we will discuss briefly on the structure of prokaryotic cell. And for this, we will draw a diagram and we will start discussing on different parts of the prokaryotic cell. So at first, the prokaryotic cell, they have a cell membrane. They have cell membrane which is semi-permeable in nature. Inside this cell membrane, there will be presence of cytoplasm. There will be presence of cytoplasm. As in previous session we have studied that in prokaryotic cells there is a no membrane bound cell structure or organelles are present. So all of the enzymes will be present in this cytoplasm. Outside this cell membrane, there will be another layer, and it is called, another layer, and it is called the cell wall of the bacteria. Cell wall of the bacteria. This is the red line I am drawing is the cell wall. It is cell wall which is made up of peptidoglycan. Okay. In some bacteria, we can also found that outside this cell wall, there will be presence of another gelatinous structure or jelly-like or gelatinous layer, and this layer is known as glycocalyx. It may be present in some bacteria and it may not be present in some bacteria. If it is present, it will be surrounded the whole cell wall. It will be present in outside the whole cell wall. So this glycocalyx layer can also be present as a thin slime-like structure, or it can be also present like a capsule-like structure. Then these three portions, the glycocalyx, the cell wall and cell membrane, these three layers together form the cell envelope. Okay, it will make the cell envelope. Then in this cytoplasm area, there will be a DNA, and this DNA will be circular. There will be a DNA and the DNA will be circular. The circular DNA is basically called, it will be a double-stranded DNA and it is circular in shape. This circular DNA is basically called genomic DNA. And in bacteria, we can found that outside this circular DNA, there will be another DNA. That is known as plasmid. That is known as plasmid. This circular DNA is called genomic DNA, and the extra chromosomal structure which we can found in bacteria is known as plasmids. In this plasmids, we can found several genes such as some resistance genes which will provide resistance against some antibiotics. There will be a presence of some F genes or a fertility gene or fertile gene, and which will help in the production of the sex pili. Then there will be presence of another DNA part of another gene which will help to protect this bacterial structure against some chemicals produced by E. coli. That is known as col gene. So the genomic DNA In general, it will be present in the middle of the cells, but at the time of cell division, it will be moved and it will present in this region or cell membrane region. The cell membrane structure in bacteria, in the cell membrane we can found some infoldings. Okay, in this bacteria we can found some infoldings. So let me just make this portion and we will draw the infoldings. The infoldings can be of tubular in shape, and these infoldings are known as, these are some infoldings, and these are known as mesosomes. In these mesosomes, we can found that there will be present some respiratory enzyme which will help the bacteria in the process of respiration, and these mesosomes will also help this bacteria in the process of cell division. It will help in the formation of cell wall and also DNA replication process, and it will also contain some respiratory enzymes which will help in the process of respiration. In the time of cell division, this circular DNA will move towards this mesosome and then the cell will be divided. As we have already studied that in this bacterial cell, we don't found any membrane bound cell organelles, except we can found only ribosomes. These are ribosomes. And these ribosomes are basically 70S in nature. These ribosomes are considered as a protein factory, or the protein will be present or produced here. Then in some cyanobacteria, we can found that, as we know that the cyanobacteria can do photosynthesis. So in some cyanobacteria, we can found that the pigments will be present in another structure, and this structure will be known as chromatophores. Okay, it will be found only in cyanobacteria. And then now we will discuss about the some outgrowth of the cell. We can found that in this bacterial cell, we can found that there will be presence of some elongated filament like structure which will be present at the outside of the cell. This is the filament like structure, and these structures will help in movement of this bacterial cell. These structures are known as, this structure is known as flagella, which will help in the movement of the bacterial cell. And this flagella has three parts: our basal body, a filament, and in this basal body and filament, between this there will be a hoop. Next, we will discuss about some other outgrowth or outer structure of this bacterial cell. So there will be present some such type of structure. Okay, these structures are known as fimbriae. These fimbriae are basically small filamentous or small bristle-like structures which we can found in bacterial cell, and we also can found that there will be some elongated structure which are have more length than these fimbriae, and these are known as pili, or they can also be called as sex pili. So we in this pili and fimbriae, this fimbriae basically helps to attach this bacteria with some substrate, and the pili will help in the process of conjugation of this bacterial cell. When we were discussing about the structures or different organelles or ribosomes, then we have also have to study that there will be some presence of some inclusions bodies in this bacterial cell. which are basically these are food materials which are present in this bacterial cell. These are called cell inclusions. So, in this session, we have studied the structure of a prokaryotic cell. We have studied that in the presence of DNA, which is not inside any nuclear membrane, because the nucleus of this prokaryotic cell are not well organized, and which we have also observed that beside this circular DNA, there will be presence of a extra chromosomal DNA part that is known as plasmid. On the basis of presence and absence of plasmids, the bacteria can be of two types. If the plasmid is present in some bacteria, then they will be considered as donor, and if it is not present, then they will be considered as recipient, or they can be considered as donor or male, and they will, if the plasmid is not present in some bacteria, they will be considered as recipient or female. And this pili will help in the conjugation of this bacteria, and fimbriae will help it to attach these bacteria with some substrate, and the flagella will help in the movement of this bacterial structure. And there will be not presence of any membrane bound organelles, there will be presence of only ribosomes, and these ribosomes will be 70S in type. So, this is the complete structure of a prokaryotic cell. Thank you.